Why is everything so confusing?
I study for hours, but nothing makes sense. Am I the only one struggling like this?
Welcome to NeuroNectar, where science finally makes sense.
Welcome to an in-depth look at one of life's most critical structures, the plasma membrane, often called the cell's security gate. To understand the plasma membrane, we must first understand the environment it protects. Have you ever wondered what it must be like inside a cell? It's not just an isolated little world. A cell has a lot going on inside, but it also constantly interacts with its environment. To stay alive and maintain a stable internal environment, a state known as homeostasis, the cell must have strict control over what goes in and out. The key structure responsible for maintaining this crucial balance is the plasma membrane. Think of the plasma membrane as a highly regulated fence or security gate, controlling all passage into and out of the cell. This membrane is composed mainly of a phospholipid bilayer, meaning it consists of two distinct layers of lipid molecules. Each phospholipid has a polar, hydrophilic, or water-loving head that faces outward, and two nonpolar, hydrophobic, or water-fearing tails that face inward, away from the water. This unique structure gives the plasma membrane its most important functional characteristic. Because of this unique sandwich-like structure, the membrane is known as selectively permeable. This means it allows certain substances to pass through easily, while strictly preventing others from crossing the boundary. Substances cross this crucial boundary in one of three main ways. The first method is passive transport, which requires no energy from the cell. The second method is active transport, which requires the cell to expend energy. And finally, we have bulk transport, used for moving very large quantities or large particles. Let's break down each of these transport mechanisms in detail. Now we will take an in-depth look at passive transport, the cell's way of moving molecules without expending energy. Our first category of movement across the cell membrane is passive transport. You can think of passive transport like riding a bicycle down a hill. You don't have to pedal because you are simply coasting downwards. In cellular terms, molecules move down their concentration gradient, flowing naturally from an area of high concentration to an area of low concentration until equilibrium is reached. Because this process goes with the natural flow, the cell does not need to expend any ATP energy. Passive transport happens in three specific ways. Number 1, simple diffusion. Small, non-polar molecules can slip right through the hydrophobic interior of the lipid bilayer without any assistance. For example, if there is a higher concentration of oxygen gas outside the cell, it naturally diffuses straight across the membrane and into the cell. Carbon dioxide easily diffuses out in the exact same way. Number 2, facilitated diffusion. Some molecules, like simple sugars, amino acids, and charged ions, are blocked by the non-polar lipid bilayer. They need help or facilitation from special transport proteins. Channel-mediated diffusion uses water-filled protein channels to let specific charged ions, like sodium, pass through. These can be either leak channels or gated channels. Carrier-mediated diffusion uses carrier proteins that physically change their shape to ferry polar molecules, like glucose, to the other side. Number 3, osmosis. This is specifically the passive movement of water. Osmosis is the passive movement of water through a selectively permeable membrane. Water moves to achieve balance, traveling from an area of higher water concentration, meaning low solute, to lower water concentration or high solute. To travel at a fast rate, water uses specialized integral protein channels called aquaporins. In summary, passive transport is the movement of molecules down their concentration gradient without requiring cellular energy. Remember the three types: simple diffusion, facilitated diffusion, and osmosis. Now we'll tackle the concept of active transport, the uphill climb. Another world of cellular mechanics. But what if the cell needs to move a substance against the natural flow? Welcome to active transport. This is like swimming upstream or pushing a bike up a steep hill. Molecules are forced from an area of low concentration to a place of higher concentration. Because this goes against the concentration gradient, the cell must expend cellular energy in the form of ATP or adenosine triphosphate. There are two main types of active transport. First, primary active transport. Primary active transport uses cellular protein pumps, often called ion pumps, to physically force ions across the membrane. The most famous example is the sodium-potassium pump. 3 sodium ions from inside the cell bind to the pump. An ATP molecule acts as fuel, and the energy causes the pump to change shape, releasing the 3 sodium ions outside. Then, 2 potassium ions bind from the outside, the pump reverts, and the potassium is released inside the cell. The second type is secondary active transport. Secondary active transport is like hitching a ride. A substance is moved against its gradient by using the energy provided by a second substance that is moving down its concentration gradient. If both substances move in the same direction, the protein is called a symporter, like sodium pulling glucose inside with it. If they move in opposite directions, it's an antiporter, like sodium moving in while hydrogen is pushed out. Now let's take an in-depth look at a critical process in cell biology, bulk transport, often known as vesicular transport. We are moving beyond simple diffusion and active transport to explore how cells handle the big stuff, the cargo ships of cellular movement. Bulk transport, or vesicular transport, is the mechanism cells use to move huge quantities of materials across the plasma membrane. Sometimes the cell needs to move huge materials, like massive proteins, large carbohydrate polysaccharides, or even whole bacteria that are simply too large to fit through any transport protein. To accomplish this, the cell membrane physically folds in or out to form membrane-bound sacs called vesicles, which act as the cell's cargo carriers. This heavy lifting process, unlike passive transport, heavily requires metabolic energy, or ATP, to change the membrane shape and move these large molecules. Bulk transport generally goes in 2 directions. First up is endocytosis. Think endo for enter. In endocytosis, the cell membrane folds inward, traps a large substance, and pinches off to form a vesicle inside the cell. It's the mechanism for internalizing materials. Phagocytosis, or cell eating, is one specific form. It involves the cell engulfing large solid particles. For example, an immune cell will engulf a bacterium, package it into a vesicle, and digest it. Pinocytosis, or cell drinking, is another form. Here, the membrane folds inward to gulp tiny droplets of surrounding interstitial fluid and any dissolved solutes it contains. Finally, receptor-mediated endocytosis is highly specific. Target molecules must first bind to specific receptor proteins on the membrane, which then triggers the membrane to fold in, enclosing the receptors and their payload. The second direction of bulk transport is exocytosis. Think exo for exit. Exocytosis is how the cell secretes materials out into the surrounding fluid, sending large molecules or waste out of the cell. Vesicles formed inside the cell, often by the Golgi apparatus, travel to the cell's border. The lipid bilayer of the vesicle seamlessly fuses with the plasma membrane, releasing its contents outside. This process is crucial for eliminating cellular waste, secreting hormones for communication, or getting large carbohydrates out of a plant cell to build a new cell wall. Bulk transport, carried out by vesicles, is vital for managing materials too big for simple channels. Remember, endocytosis brings large materials in, while exocytosis secretes them out. Understanding bulk transport helps us appreciate the cell's remarkable ability to manage its massive traffic flow, essential for life, defense, and growth. Now we're diving into a quick summary of how the cell membrane manages transport, a vital process for all life. The cell membrane is constantly working to maintain the perfect internal environment. Let's recap the 3 incredible ways it moves materials in and out. Get ready for a visual journey through passive, active, and bulk transport. First, let's remember the membrane's crucial role. It is the ultimate gatekeeper, deciding exactly what enters and leaves the cell. This lipid bilayer acts as a selective barrier, ensuring the cell maintains its necessary internal balance. Our first method is passive transport, which is all about effortless movement, or coasting downhill. Think of it as simply sliding through the membrane, like an oxygen molecule. No energy needed. The key principle here is movement down the concentration gradient, from high concentration to low, meaning no energy is spent. Next up is active transport, the method for pushing uphill against the natural flow. This requires specialized protein pumps that physically push molecules against the current, much like swimming upstream. Since this goes against the gradient, the cell must pay an energy cost, primarily in the form of ATP. Finally, for the largest cargo, the cell uses bulk transport, essentially shipping massive cargo. This is how large quantities or entire objects are transported, by wrapping them in membrane packages called vesicles. Whether bringing things in via endocytosis or pushing them out via exocytosis, vesicles are the cell's essential shipping containers. So why do all these transport methods matter so much? It's about life itself. Together, these incredible, continuous microscopic systems keep our cells functioning, ensuring everything from nerve impulses to digestion works perfectly.